Welcome to this webinar titled Reducing Time to Manufacturing Using Real-Time In Situ FTIR Analytics as a PAT Tool. We all share the goal of developing a better synthetic route or chemical process quickly, efficiently, and safely. The chemists and engineers that we work with around the world in the chemical and pharmaceutical industries, and also in academia, have told us that this is their clear goal. We work with scientists as they try to leverage the use of modern lab instruments like automated lab reactors and real-time analytics to develop greener synthetic routes, more robust, reliable, and efficient chemical processes, and safer scale-up conditions. Today's webinar will focus on the use of in situ FTIR to provide critical information during drug development that will enable the reduction of time to manufacturing via better process understanding and, in some cases, feedback control. Here is a brief outline of what we will cover today. We will start with an introduction into some of the concepts behind the FDA's Quality by Design (QBD) approach, incorporating process analytical technologies (PAT). I will also briefly describe the relevance of FTIR as a PAT tool. I will then review some examples from the literature where FTIR has provided the necessary information to move projects forward to a larger and larger scales, thus reducing the time to manufacturing. Although the selected examples come from the pharmaceutical industry, the use of in situ FTIR, of course, applies to a wide variety of chemical reactions. Indeed, the chemical, specialty chemical, and polymer industries, to name a few, utilize this technology routinely during development and on into production. While reviewing the publications, I will do my best to capture the essence of each project and the role that the in situ monitoring played in supporting the project goals. For full details, please refer to the original article, which will be listed at the bottom of each slide. After a brief summary, I'll be happy to answer any questions that may arise regarding the information presented. In other industries, government oversight and regulation is not quite as keen as is found with pharmaceuticals. Since 2002, the FDA's Guidance on Process Analytical Technology, PAT, has had a significant impact on the way potential medicines are discovered and developed. The guidance is also influencing NDA filing methods at many pharmaceutical companies. The change has been fairly gradual overall and is most prominent in the largest pharmaceutical companies. However, all sizes of companies are investigating and applying the growing range of PAT tools. The latest FDA guidance for industry, released in November 2009, defines quality by design as, quote, a systematic approach to development that begins with predefined objectives and emphasizes product and process understanding and process control based on sound science and quality risk management, unquote. A process analytical technology is defined as, quote, a system for designing, analyzing, and controlling manufacturing through timely measurements, i.e. during processing, of critical quality and performance attributes of raw and in-process materials and processes with the goal of ensuring final product quality, end quote. I trust that most of you are familiar with the general goals of PAT. First, to understand the process by thoroughly mapping the design space and understanding the critical quality attributes, CQAs, of a given product, and learning what critical process parameters directly affect product quality. Understanding this provides the basis for a risk-based quality by design. Second, to predict product quality in real time through monitoring of critical process parameters using appropriate process analytical technology, which provides the basis for quality assurance and real-time release. And third, to eventually provide the ability to control the process to assure a, quote, right first time, unquote, production, avoiding batch failures and costly rework. How can PAT help? While the general benefits of PAT still apply, successful implementation of PAT and QBD should result in a better overall process with less upsets, product variability, or batch failures. This can certainly reduce the cost of root cause analysis and regulatory review for process and product quality deviations. Additionally, PAT and data-rich experiments can certainly enable the process design specialist to better understand the process and be able to reach an optimized process faster, reducing the amount of time and resources spent on developing each process. This can be especially important during a project or technology transfer between sites or between companies. Using PAT early on in development can be especially valuable since the knowledge gained can save valuable time down the line. I have certainly observed in the past 10 years the use of FTIR earlier and earlier in drug development and its use as a more routine tool. Follow-on benefits to the improved research and development capabilities are simply the benefits of a better process design, more reproducible production, better product quality, better resource utilization by eliminating bottlenecks in the pilot plant or the production facility. Ultimately, this should all lead to less waste, higher productivity, better medicines for patients, and higher profits for companies. On top of that, PAT technology provides clear data, a highly effective means of communicating exactly how the process is expected to perform and exactly what, in terms of the critical process parameters, affect product quality. This clarity of not just product specs, but process parameters enables the organizations with PAT capabilities to deliver results that meet expectations quickly and profitably while speeding the time to manufacturing. So why is real-time reaction monitoring using a PAT tool so useful? The objective in using real-time PAT is to answer many of the same questions when a grab sample is taken and run an offline analytical method. However, the real-time nature does a better job at connecting all the dots typically obtained using sampling for offline analysis due to the much higher Templar resolution. For example, a spectrum collected every minute. No critical information or subtle process behavior is missed at the beginning, in the middle, or at the end of the reaction. Also, real-time analytical technologies do not require waiting for the answer as you commonly have to when using an offline method, which can result in missing the optimal endpoint. In some cases, errors associated with a quench during sample preparation can be eliminated entirely. In other cases, one can interrogate species that are not amenable to offline analysis, such as molecules that do not have a UV absorbance. Very often, use of a PAT tool simplifies a chemist's life because it doesn't necessarily require the development of a dedicated method, and it doesn't require the taking of samples when it is challenging or dangerous to do so. FTIR lends itself very well as a PAT tool for real-time reaction monitoring because of the functional group specificity provided by mid-infrared spectroscopy. One can even collect a spectrum of the reaction mixture as fast as every second if appropriate. A typical experiment yields a 3D surface that shows which functional group is disappearing and what's being formed without ever having to actually remove a sample or interfere with the reaction in any way. In other words, one can profile specific functional groups in a molecule or many molecules within a mixture and can obtain concentration trends as a function of time. This is very powerful for reaction understanding and optimization. The result is a sort of movie of the chemical reaction that records the making and breaking of chemical bonds. The essence of the FTIR is the ATR, Attenuated Total Reflectance Probe. This has been reviewed before in numerous other webinars, and I will not review it here. Please follow the link at the bottom regarding FTIR method of measurement if you're interested. Essentially, the ATR probe is sensitive to the liquid phase of the reaction mixture and is not susceptible to interferences from solids or bubbles in a well-mixed reactor. The first example comes from a group of researchers at Pfizer in La Jolla, California, USA. It involves the process development and scale-up of AG035029, which was being investigated for the treatment of type 2 diabetes. Material demand for this molecule escalated very quickly over a short period of time, such that greater than 10 kilograms of drug substance was needed to support toxicity in clinical trials. In order to provide this amount of material, the medicinal chemistry route was reevaluated. It was a linear route that consisted of an expensive material and contained a step that could lead to high-energy compounds. The challenge to the process R&D team was to develop a new convergent route that was rugged and capable of delivering product with reliable quality. Of course, all of this must be accomplished quickly and safely with reasonable cost. The convergent route shown in the upper right was proposed. Upon the first attempt, a few issues were identified. The nitro reduction in the first step was not totally selective, and some debenzylation was also observed. A further catalyst screening study was undertaken, where 3% platinum on carbon provided an 80% yield with no debenzylation. A second and much bigger problem was observed during the Michael addition to add the N1 side chain. Significant double addition was observed and produced up to 25% of compound 16, as shown in the bottom right. This byproduct proved extremely difficult to remove, even via chromatographic methods, crystallization, or running at lower temperature. The problem got worse upon scale-up, with the levels of the byproduct increasing even further. Changing to 3-bromopropionate as the N-alkylating agent did not result in the undesired double Michael addition product 16. However, a mixture of isomers 14 and 17 was now obtained. Alarmingly, this process was capricious in that sometimes the desired product 14 would be obtained exclusively, while other times up to 20% of the undesired isomer 17 would be produced under what were perceived to be identical reaction conditions. As the authors state, quote, "The reaction was clearly not fully understood, and hence was out of control. Experience has shown that such processes are prime candidates for study with FTIR." Which immediately revealed the participation of an intermediate species absorbing at 1549 wave numbers, ruling out a simple SN2-like mechanism, unquote. The reaction was also studied with FTIR using toluene as the solvent with no DMF present. In this case, no intermediate was observed with the FTIR. A slight induction was observed at 80 degrees C, while the reaction at 90 degrees C displayed nth-order kinetics, typical of an SN2 type reaction. One important observation was that there was always poor selectivity when neat toluene solvent was used. This seemed to imply that DMF is quite important and that a different mechanism was at work. The intermediate observed in toluene DMF must be the key to selectivity of this reaction. The authors suggest a possible mechanism and use that model to provide simulated species profiles to compare with the observed FTIR profiles, as shown on the right. A final FTIR experiment unlocked the key for this reaction and its quick scale-up. The mixture was slowly heated from room temperature to 90 degrees C. The IR peak, due to indazole starting material, started to decrease at 30 degrees C and was completely gone at 40 degrees C, resulting in the intermediate species. Continued slow heating revealed that the intermediate began to decrease once the reactor exceeded 60 degrees C to provide the desired product 14. With the information provided by FTIR, the solution to the capricious behavior was clear: an isothermal hold at 40 degrees C to allow for complete formation of the intermediate species, followed by heating to 90 degrees C to provide the desired product 14 via collapse of the intermediate. The authors indicate that following this methodology, the process was highly reproducible and was successfully scaled up to a greater than 20 kilogram level in the Pfizer pilot plant. It is worthwhile to note the use of FTIR as a PAT tool in the lab provided the necessary process understanding to design a robust process that did not require the use of a PAT tool in the pilot plant. Additionally, PAT tools utilized in this study included parallel lab reactors, computational chemistry for simulation, and FBRM probes for crystallization development. The next example comes from researchers at Merck, located in Hertfordshire, UK, and Rahway, New Jersey in the USA. Pyrazole 1 was targeted for development due to its potential use as a nicotinic acid receptor agonist for the treatment of arteriosclerosis. Due to material demand pressure, the goal was to exploit the existing medicinal chemistry route in order to quickly supply bulk drug quantities for toxicology and clinical studies. However, several challenges were quickly identified in the medicinal chemistry route that needed to be addressed, namely the use of high pressure, greater than 200 psi, in the formation of amide 5, the need for protection and deprotection steps, and the sodium azide addition at elevated temperature of 120 degrees C. The modified route is shown on the bottom right. The amide 5 was formed via treatment of acid 8 with 1,1-carbonyl diimidazole, CDI, thus avoiding the use of high pressure. The need for benzyl protection was avoided by changing to milder dehydration conditions using trifluoroacetic anhydride, TFAA, and triethylamine in THF to yield the nitrile 9. Treatment of the nitrile with sodium azide would then give the desired tetrazole 1. It was found that the tetrazole could be obtained in good yield, about 83%, in approximately 10 hours at 75 degrees C versus the 120 degrees C in the medicinal chemistry route. Additionally, hazards evaluation showed no undesired exotherm during the sodium azide addition. The hazards evaluation also included the use of a heated infrared gas cell attached to a FTIR 4000 system with which to analyze the reactor headspace. The gas cell is shown in the picture on the bottom left. Analysis of the headspace during the sodium azide addition showed the presence of hydrozoic acid, a volatile, highly toxic compound that also is potentially explosive in the condensed phase. The figure at the bottom right shows gas phase reference spectra of the solvent, 2-propanol, carbon dioxide, and hydrozoic acid. The sharp mid-infrared peaks in the gas phase made it quite easy to differentiate all these species. A plot of these three species with time during the azide addition is shown in the top right. Unfortunately, no process modifications could be found that would satisfactorily reduce the risk of this hydrozoic acid release. Thus, the azide reaction was deemed too hazardous to scale up, and the modified medicinal chemistry route was abandoned. This necessitated development of an entirely new route to provide the needed quantities of the tetrazole in a timely manner. Sometimes having to start over again can yield an even better approach. Such is the case here with the resulting method shown in the upper right. It is a one-pot process that starts with a commercially available tetrazole ester 10. This process is significantly more streamlined than the 7-step medicinal chemistry route that yielded 39% of the desired product. The one-pot process represents a viable 2-step manufacturing route while increasing the overall yield to 90%. The authors indicate that this process was successfully performed at the 70 kilogram scale without incident. The next example comes from researchers at Bristol-Myers Squibb, located in New Brunswick, New Jersey, USA. It describes the multi-kilogram preparation of ravuconazole, which was of interest as a broad-spectrum antifungal agent. The references describe the evolution of the process through 3 campaigns, which increased in scale and sophistication as demand for the drug substance continued to increase as the molecule proceeded through the pipeline. One key intermediate in the synthesis of ravuconazole was the aryl-propanone 7, as shown in the bottom right. The formation of Grignard reagent 6 initiated reproducibly in the lab and performed adequately up to the less than 1 kilogram scale. The first campaign made use of the kilolab facilities, but in order to supply batches of 10 to 100 kilograms, which were anticipated for the next campaign, significant changes in the operational procedure were deemed necessary. Grignard reagents are well known for their synthetic utility and are also well known for their challenges upon scale-up. These include irreproducible or undetectable initiation, difficulty in tracking the formation of the Grignard reagent, dangers in handling the pyrophoric metal turnings, and the hazards and challenges associated with controlling a very exothermic reaction. In this case, the metal insertion was 380 kilojoules per mole. The researchers did investigate a Barbier modification in which the Grignard reagent 6 would be prepared in the presence of the electrophile 4. This would alleviate the stability issues associated with storing the Grignard reagent. Unfortunately, this approach provided unsatisfactory results, and the traditional 2-step route had to be used. The Process Safety and Evaluation Group determined that the aryl bromide concentration in the reactor should not exceed 15% of the total charge at any time during the feed, such that the adiabatic temperature rise not exceed 33 degrees C. FTIR proved to be a critical tool for process monitoring of this reaction in that each of the species of interest could be tracked: the aerobromide 5, the Grignard reagent 6, starting material 4, and formation of the coupled intermediate 18. Although useful for process understanding in the lab, it was deemed very important to incorporate in situ FTIR at scale in the pilot plant. FTIR was found to be real-time and quantitative for all the species mentioned, negating the need for slow offline sample analysis. A key aspect of utilizing FTIR in the pilot plant would be detecting reaction initiation and ensuring a proper feed rate of the aerobromide while avoiding any reaction stalling. Feedback from the mid-infrared PAT tool allowed the addition rate to simply be decreased if the aerobromide addition approached the predetermined safety threshold. Once the Grignard was formed, the amide 4 was metered in. Again, FTIR provided real-time feedback as shown by the disappearance of the Grignard signal in pink at 1582 wave numbers. The use of FTIR helped to safely and reproducibly afford 5 pilot plant batches. A short note regarding the implementation of FTIR in the pilot plant. In this case, a recirculation loop was employed with a 1-inch diameter Sentinel die count probe. The FTIR instrument used was rated for Class 1 Division 1 operation. When working at a larger scale, as found in the pilot plant or manufacturing facility, there are a variety of ways to incorporate a FTIR probe. Of course, it always depends upon the particular reactor suite and the application. These topics have been addressed in previous webinars. If you have questions regarding your specific situation, a METTLER TOLEDO Technology and Application Consultant would be happy to discuss them to provide the best advice. This last example comes from researchers at Johnson & Johnson in Beers, Belgium. A key step in the synthesis of an estrogen receptor modulator involved a 1-carbon homologation via an enolate intermediate. Deprotection of 1A with lithium hexamethyldisilazide, LiHMDS, yielded the enolate, which was then quenched with POMCl. It was quickly observed that the substrate to base ratio was critical to optimal reaction performance. If too much base was added, impurities shown in the bottom right could be formed. If the base was undercharged, it resulted in lower conversion and the need for a recrystallization step to remove excess starting material, which led to further product loss. Clearly, this reaction needs just the right amount of base, which would be difficult to assess via offline analysis since the intermediate is moisture sensitive. An in situ PAT tool would be perfect for this need. FTIR was incorporated to further understand this step and ensure a robust process. As shown in the plot, FTIR reveals clear bands at 1726 wave number due to the starting material and at 1625 wave number due to the enolate. Having a tool with which to monitor this step would be crucial at larger scales to avoid batch-to-batch variation due to the varying starting material quality. It was also noted that on small 20 milliliter experiments monitored with FTIR, 1.2 equivalents of base were needed versus 1.02 equivalents when run on slightly larger scale. The authors attribute this to different moisture conditions found at the two scales, and state this as yet another reason to monitor this deprotonation step with a PAT tool. One could not simply add a predetermined amount of base. The deprotection of every batch needed to be monitored to ensure consistent reaction performance. The figure on the top right shows the enolate peak at 1625 wave number increase during the base addition and the carbonyl peak at 726 wave numbers decrease. This was performed at the 5-liter scale. The figure at the bottom right shows the next step, the addition of the enolate to a solution of POMCl. In this case, the carbonyl peak at 1756 wave numbers from POMCl is shown to decrease as it is consumed, and a peak at 1726 wave numbers from the end product forms. This reaction was also run in DMF, which added a little twist to the spectral interpretation. DMF has a fairly strong infrared band in the carbonyl region, which slightly masks the smaller carbonyl from the starting material. However, a simple spectral data treatment of applying a second derivative allowed the 1725 wave number peak to still be monitored. This second derivative is a standard data treatment within the ICIR software package. Through the use of ReactIR, several key points were uncovered. 1. The IR spectrum of the Enolate remained unchanged for up to 4 hours at room temperature under inert conditions. Obviously, this makes handling at larger scale much easier. 2. A slight excess of base, 2 to 4%, gave the best results. This differed from a previous report for a deprotonation step by Watson et al. At BMS, where a slight excess of starting material performed best. The first body of that work was published in the Journal of Organic Process Research and Development in 2004 and was titled Development of a Safe and Scalable Oxidation Process for the Preparation of 6-hydroxybuspirone. It was also presented at the METTLER TOLEDO Real-Time Analytics Forum in London, UK in the same year. The second body of this work was published in 2008, again in OPRD, and was titled Development and Scale-Up of 3 Consecutive Continuous Reactions for Production of 6-hydroxybuspirone. That batch and continuous process was also the subject of a recent METTLER TOLEDO webinar entitled Enhanced Development of Batch and Continuous Processes Using Real-Time In Situ FTIR Analytics as a PAT tool. With the ability to determine the base endpoint, information regarding the kinetics of the deprotonation and the quench, and knowing the stability of the intermediate, the scale-up of this reaction proceeded smoothly and was uneventful. I would like to conclude by making some observations about the case studies presented here, and based upon my many years of experience. As we learned from the researchers at Pfizer, use of ReactIR as a PAT tool provided the necessary reaction understanding with which the proper reaction parameters could be defined in order to yield a reproducible and well-behaved process upon scale-up. In this case, the PAT tool itself did not need to be utilized at the larger scale. This might seem a bit surprising, but is really quite common. The example from Merck highlights an aspect of ReactIR that you might not have thought about, the use of an additional accessory for an existing ReactIR system. The modularity of sampling options allows the ReactIR to be quickly adapted for a given task, be that a fiber optic probe, a gas cell for headspace analysis, a microflow cell for continuous processes, or a sentinel probe for monitoring reactions under pressure inside a PAR reactor. Take advantage of an existing tool by keeping the software up to date and adding on sampling technology. In my experience, most mid-sized to large-sized pharmaceutical companies have access to a ReactIR in the Kilo Lab. However, their use has been somewhat sporadic depending upon the project. It is becoming more and more routine to use this tool as a fingerprint regarding the batches and saving the recorded moving of the process. If issues are discovered downstream, such as in another step, or another unit operation like a crystallization, or further on into formulation, there may be useful information to be gained from the ReactIR dataset with which to help solve the problem. Of course, in certain cases, such as that described in the Grignard example from BMS, the tool was used for feedback control due to safety concerns at the pilot plant scale. Companies that have invested in these PAT tools are clearly reaping the benefits afforded through their continued use. Finally, I would like to encourage the routine use of ReactIR as a PAT tool. Don't wait for a problem to present itself before using these tools. The current generation of ReactIR with flexible fiber optic conduit probes makes monitoring a reaction as easy as inserting a thermocouple, and I can assure you that oftentimes one will learn something new regarding reactions you thought you already knew everything about. This process understanding is a key aspect in reducing the time to manufacturing in today's challenging times.